Hello everyone. So today I will be teaching about. I won't start any topic or something since this is the last class. This is week 10 of my TA-ship in week 12. So I will be today teaching about uh, everything. Like I will just go through the basics from first slide to last slide since your exam is also coming. So I will quickly glance through the slides and I will uh, teach about teach third slide only. So there is a quick revision for every one of you. So first we will I will I hope everyone can see. So I'll be teaching about from the beginning, that is history of immunology. So we will quickly glance through the thing like history of immunology 1. This is first we know birth of immunology. The word immunity means immunis, which means exempt in Latin. So first in the post 30 BC, historian Thucydides in his contemporary, he described about the plague. Then from that time, it was known like uh, that something called immune system is there in every people, and so whoever is affected by a disease, in that time it was plague. So that time, that same person is not attacked twice, or that same person is not affected by twice by the same disease. So it first began with the smallpox disease, uh, which claimed so many lives in Europe, and it was like it thousands of years ago it emerged and it caused many illness and deaths in human populations. The smallpox is having a linear double-stranded DNA genome that vary from 150 to 230 kbp, and the symptoms include high fever, a headache and body ache, sometimes vomiting, disfigurement, and death. And by there is like uh, blisters happening on your skin, and there will be fluid inside the uh, source, and they will become pustules, and then after that uh, they will be dry. So it was like in the beginning, like in 864 to 930 AD, it was Al-Razes, of Persian physician, who explained that smallpox and measles was a disease. He also explained about the allergy for the first time. So smallpox appears when blood boils and is infected, resulting in vapors being expelled. So we all already have seen this about the smallpox. So what happened? There were different kind of practices which followed to prevent the smallpox. In India, it was done using a sharp iron needle where they were pricking through the pustules, and then usually in the upper arm they punctured the skin. And in China, it was happening like based on blowing like smallpox material up the nose of the child who was inoculated. Sometimes they will put dry scabs into powder, and other times they will extract it into water. And sometimes they will collect the fluid from a pustule and place that up the nose. So there comes a term known as variolation, which was in 1714. It was reported as in the Royal Society. It was the first immunological clinical trial for smallpox variolation. Variolation means spotted or stain. So what they were doing, they were putting the fluid from the pustules into the individual, so they are protected from the disease. So first ever vaccination occurred by Edward Jenner. It was known as vaccination. because it was a word derived from vaxa, which means cow. So there was global eradication of smallpox by 1979. So what happened was, there was exposure to cowpox, which protected the individual against the more dangerous smallpox. So from the cowpox, they started taking that same fluid and injected into the people. Then they were saved from the smallpox. So it was kind of vaccination that time. Then first modern controlled experiment in immunology was done by Louis Pasteur, who is considered as the father of immunology. So he first vaccinated one group of sheep with some heat killed or heat attenuated bacteria. Then he challenged that vaccinated sheep and some unvaccinated sheep with a virulent culture of the bacillus, and and that all the vaccinated sheep lived. However, the unvaccinated animals obviously died. It was kind of a first experiment which was done by Louis Pasteur, to which we follow to today, like what we call as vaccine. That time it was done by him. So he did this to a person, first vaccine to a young boy who was repeatedly bitten by rabid dog. So to prevent rabies, he administered this first vaccine to that boy. Then comes some postulates given by Koch, that there are some postulates like around 1875 by Koch, which showed that he stated that there are microorganisms which are found in the diseased individual. Like whenever there is a disease in some person, then some microorganism might cause it. But in healthy individuals, we do not find these microorganisms, and we can collect that microorganism from the diseased individual. Then we can inoculate that healthy individual with some microorganism from a diseased individual. So when we are inoculating, that disease might occur in that healthy individual, and then again we can, from that diseased individual, we can again take the uh, like the microorganism back, and we can match it to the original organism. So Scotch, Robert Koch, he also discovered the organism Mycobacterium tuberculosis, which causes white death or the tuberculosis, and he also showed a test which is a tuberculosis skin test, which is still followed. So it he showed that if he injects like tuberculin, which is a culture or a product of the bacillus or the bacteria, it will create several inflammatory reaction in tuberculous patients upon when they are applying to the skin. So it test has proved to be invaluable even today, as I told. As last class also I told, tuberculin skin test is still being followed, where you can detect whether a person is having TB or not. And so then came the birth of immunology. As I told, smallpox was the starting. Then Edward Jenner obviously uh, put his first vaccine. Then there is different ways of variation in different parts of the world. So then came two types of a dichotomous thinking in immunology. There were two groups in two immunologists, like cellularists and humoral. So the cellularist used to tell that this immune system, this immune power is given by some cells, which specifically comprise of this macrophage or the macrophage and the microphage, which are the leukocytes. And this human means some body fluid. So this they used to tell that it is caused by the humor or the something which is inside the body fluid. So there are two groups. For example, this Eli Mechnikov, then Charles Darwin, Louis Pasteur, Robert Koch. These are the mostly main uh, people who used to get divided into two groups. And after that, uh, there were other people like, as I already told, Mechnikov, who showed that cells rather than serum components are the major effector of immunity. What he did was he placed some thorns in starfish larvae, the amoeba cellular entities that seem to engulf the thorns. Mechnikov termed such cells as phagocytes, giving rise to the field of cellular immunology. So you can understand 1884 means this much long time. Then there were people who were Paul Ehrlich, and he had these are the drawings of the formation and effector functions of antibody according to side chain theory. So he was a chemist, and in 1897 he has uh, put some theories. So he showed that there are certain toxin molecules which can bind with the antitoxins, and there are some haptophore groups using what they will bind to these antitoxins. Then cells which are under the threat of this foreign microorganism can grow the side chains to capture the toxin elements. There are additional side chains designed to break off into circulating body fluid and identified as antibodies. And the first model to propose for an antibody molecule in which the antibody was branched and considered multiple sites for binding to foreign material was known as antigen, and for the activation of the complement pathway. So basically, he just told about the antigen antibody interaction in 1897. In 1888, there were German pathologists Klebs and Loeffler who had isolated the diphtheria bacillus and demonstrated that it caused the illness. Yersin and Rokes found something very interesting, that bacteria is not entering into the blood's victim's blood, but it is producing something which is a poison, which is the cause of the disease. So this was the isolation of the diphtheria toxin. Then there is bactericidal power of blood. This is George Nuttall, and this is Edward Buchner, who also is George Nuttall. He um, found that the uh, blood which is lacking this fibrin had a considerable bactericidal power against Anthrax bacilli, and when you are heating this to 55 degree, it is getting destroyed. So the blood contains something which is actually destroying the bacteria. And here Buchner was one of the first to know that a substance in blood serum is there, which is known as alexin, can actually destroy the bacteria. Then there is Pfeffer phenomena, which is known as immune bacteriolysis. That is, the vibrios inoculated into the peritoneal cavity of normal animals rapidly multiply by the same vibrio in an animal that had recovered from the infection and underwent bacteriolysis. That is, they are rapidly swelling up, dissolved, and disappearing. So, how this is happening? So, there is something happening inside the body which is killing this bacteria. So, there must be something present in the serum of an animal uh, which is producing the same lysis in the test tube. So, this is known as the Pfeffer phenomena, and it is still in use for differentiating closely allied species. Then, then Jules Bordet, who discovered complement, which is destroyed by heating to 55 degree. Then there is like serum as a protection matrix. In 1890, it was uh, by Emil von Behring, who uses a syringe to inject a guinea pig held by lab assistant. First example, it was of prophylactic serum therapy, and he also received the Nobel Prize. He termed the serum as antitoxin. So this experiment was done by him. where in experiment A he took one guinea pig, then from from a healthy unexposed animal he took the serum and put in guinea pig 2. Then he injected with that animal with some bacteria strain diphtheria. So it happened that animal got diphtheria, but in experimental B he took some toxin from bacterial strain A. Then he what he did was he took serum from another animal which was again exposed to bacterial strain A. Then he injected it into the guinea pig. The guinea pig survived. There was no diphtheria. In case of experiment C, he again took a dose from a bacterial strain B, then serum from animal to B. Serum was exposed to B toxin, but then he injected all this animal with a toxin. So what happened? Since he put the serum from a B toxin, all his immune system will be specific to the B toxin and not the A toxin. So when he injected the animals with the A toxin, the animal had diphtheria. So there was bacterial agglutination, means their bacteria will clump together in the serum in ways specific to or determined by each serum. So it will form a clump-like structure. Then there are some experiments performed by Gruber. Uh, then some French physician Georges Fernand Weidel developed a agglutination test for typhoid, thereafter known as the Gruber-Weidel test or the Gruber-Weidel reaction. Then these are the people who won Nobel prizes in the field of immunology. So many people you can see, and they all got different prizes for different for their work. Mm, then we come to the, as we all saw, they are the uh, Nobel Prize winners. Then there we come to the different branches of immunology. There are different branches of immunology, and there are specialization as well as specialization among the branches of immunology. First one is clinical immunology, with deals with the diagnosis. If a particular disease is happening, what is the treatment, or how do you recognize that a disease is happening? This branch deals with that. Then there are certain diseases which are immunodeficiency, means the immune system is not working properly, so there will be suppressed immune response, or can be autoimmune disease, which means that your immune system is too much active, so it will be excessive immune response. So there should be a balance between the both, like there shouldn't be excessive immunity, there shouldn't be less immunity. So then cellular molecular immunology, which deals with the cells of, and the molecules of the immune system, and how they are involved in recognition and elimination of the pathogens. Then comes the cancer immunology or the oncoimmunology, which can understand the immune system's function during cancer progression and development. Then there is transplantation immunology, like whenever you are putting organ or some kind of tissue transplant is happening from one person to another. then what is happening inside the system. So autologous means from the same person you are transferring a transplant or a graft from the same person into, and then there is syngenic means from another person of a related person you are transferring. Then there is allogenic, that is a different person you are transferring. There is xenogenic means from a different species itself. Then there is development and comparative immunology. Then it deals with how there is immunity is getting developed or with the course of time, and how it is different among different species. So also how it is evolutionary conserved. So you can have many kinds of models which you can study to understand about the immune system mechanism. So there are also immunoinformatics, so computational immunology, which deals with computers, which will have certain algorithms, certain interpretation, or you can make some model to identify, or you can predict or analyze some immunological data. And mucosal immunology means it deals with the study of the immune system at the mucosal surface, the urogenital tract, and the respiratory system. In immunogenetics, it deals with between immunogenetics and immunology. The genetic variants of certain regions in the individuals can lead to dysregulation and susceptibility to certain diseases. In neuroimmunology, obviously deals with your brain, how the immune system is affecting the nervous system development, the homeostasis, and how responds to immune injuries. The structural means obviously it's structures of the immune molecules, including antigens, antibody, receptors, etc. Then there is reproductive immunology, like during pregnancy, how your immune system is changing, and during like the pregnancy, during birth, before birth, how a body's immunity system is developing, etc. Then there is nutritional immunology, which deals with the understanding the influence of nutrition on immunology. And then there is systems immunology, which deals with the system level approach. So you can study one particular system and uh, how different kinds of cells function differently in our body. Then there is viral immunology, which deals with basically host pathogen interaction. So these are the branches of immunology which we studied earlier. So again, so there are different kind of hallmarks or properties of immunities. Like there are like in our case, there are whenever there is an entry of any pathogen, our immune system will get activated. The different sizes of different kinds of pathogens are there, which can enter into our body, from the tiniest one to bigger one. So there is diversity in all these things. Like there will be chemical composition will differ. There will be various complex of biomolecules. There will be weight variation, huge size. There will be specificity. For example, LPS we get from gram-negative bacteria. From gram-positive bacteria we get flagellin. Then from fungi we get beta glucan, etc. So these are the different kinds of uh, viruses which infect our body, and here you can see like how much persistent of immunity one particular virus can create. For example, in the first case, you can see chikungunya. It can uh, lead to the production of antibodies which can stay for 30 years. Then there is discrimination among self, non-self, and modified self. It is very much important. As I told, there shouldn't be excessive immunity, there shouldn't be less immunity. So it sometimes happens that our body attack our own cells, and so it might lead to autoimmune disease. Auto means own, and there are, when our own immune system is attacking our own cells, then it is autoimmune disease. Then apart from that, there are modified cell means there are cancer cells which are modified. So it is very much important to distinguish between our modified cell and non-cell cells. Otherwise, it can lead to the attacking of our own cells, which may lead to various diseases. Then there should be tight regulation, like there will be transcription as well as translation regulation. There will be positive or negative feedback. There will be pro or anti secretion. Cellular level includes suppressor cells, example M1, M2 macrophage or T regulatory cells. So there are two types of immune system present in our body: innate immune system as well as adaptive immune system, and there is development of immune response upon activation of these two, which can lead to the elimination of the pathogens. How different arms of immunity system can talk? First, to the cell components, that is the non-immune and the immune cells, which include some pattern recognition receptors, which recognize these pathogen-associated molecular patterns, which induce inflammation, type 1, type 3 interferons, and activate the innate immune responses and initiate the specific adaptive immune responses. Uh, then we come to the different kinds of immune organs. Hi, Lakshmi, if you are having any doubt regarding the any queries or assignment, you can tell me. Okay. So now we come to the different kinds of organs of immune system. There are various organs. First is primary. There are first is lymphoid organs, which is the site for generation, maturation, and the initiation of the adaptive immune responses. Then there are primary as well as secondary lymphoid organs. The primary lymphoid organs contains immunocompetent cells, which are generated and they mature there, and secondary lymphoid organs consists of organs where interaction of antigen antibody takes place, or antigen naive immune cells takes place. Like when the antigen comes, these cells recognize these antigens and they proliferate and become more and more in number. They are present in mainly the lymphatic vessels, and they are region lymphocyte and macrophages. Then these are the organs. You can see how these are the sites where of this primary lymphoid organs. Like uh, these are the prenatal one, means before birth and after birth. You can see here embryo yolk sac, 1 week of development is the yolk sac. Then third month fetal liver looks like this. Then spleen. And after birth, it is the bone marrow. Then the central or the primary other primary lymphoid organs are the thymus and the bone marrow, where this B cell and T cell gets generated and mature. Then there are secondary lymphoid organs, which are the lymph nodes, spleen, and mucosal, and you can see these are the sites where these are found. So, So thymus, as I told, is a primary lymphoid organ. It is situated on the top of the heart. It's pinkish gray in color. You can see this thymus. It is the region where 99%, 95 to 95, 99% of T cells die, and the there is clonal selection and clonal deletion. Uh, if there is self reactivity, there is clonal deletion, when or no reactivity to antigen, and if there is normal, like when there is a when antigen is there and it is recognized by the cells, then there is a clonal proliferation or selection. This is the cross section of the thymus, which consists of medulla and the cortex. Uh, This cortex contains this T cells, and medulla is not densely packed. There are different kinds of immune cells found, like disease macrophage epithelial cells, and there you can remove this thymus from your body. If you remove, what happens? There is a dramatic decrease of the T cells. It is found in a syndrome which is known as human Digeot syndrome. So bone marrow is a place where there is site of maturation of B cell generation, and with other type like fat cell, and with
Ma'am, I have a
advanced.
general doubt. So
Yeah,
in
yeah.
blood cancer, in blood cancer and all, they sometimes have heard about bone marrow transplantation.
Yeah,
So,
yeah.
so the person with leukemia means what happens? Like, what are they doing with that bone marrow? Do they like completely change the blood flow or something like that? Actually, I
Bone
didn't understand that concept. That's why I'm asking.
bone marrow transplantation means, like, you will take from a healthy person some blood forming stem cells, you will take, and you will give it to a person who is
Okay.
diseased.
Hmm.
So that stem cells, if you are giving, it will produce more and more number of your like the T cells or
RBCs.
B cell,
Hmm. Hmm.
everything, but in a healthy perspective, not from the disease perspective. So,
Okay.
so it
Hmm.
will be all the cells which are produced will be healthy,
Hmm.
not the cancer cells. Like if it is a blood cancer, then they won't produce the cancer cells.
Okay.
Huh. So bone marrow is a site of B cell generation, and it is mainly filled with fat with increasing age. And here you can see this is the process of maturation, then antigen stimulation, clonal proliferation. This is like first the mother cell is the hematopoietic stem cell, then there it becomes in the bone marrow. All this is happening like lymphoid cell. There will be a pro B cell where there will be, as I already told, B in B cell there will be like heavy chain and light chain arrangement. So first heavy chain will arrange, then there will be pro B cell, then there will be complete heavy chain arrangement. Only then it will be known as T B cell. Then there are different kinds of antibodies, which are five kinds we all know. So here mu means IgM, and there will be two types of light chain is there. One is kappa and one is lambda. So then after heavy chain rearrangement, there will be light chain rearrangement. Then it will form a immature B cell. Then there will be RNA processing, and it will be forming a mature B cell. So then there in the peripheral lymphoid organs, this B cell will be inactivated. But whenever there is some kind of pathogen entering into our body, there will be some antigen which will meet these B cells in our lymphoid organs and will activate the B cell, which will result in the differentiation and proliferation of these B cells. So there will be plasma cells formation of these plasma cells which will secrete these antibodies. There are other phenomena like class switching, where apart from IgM there will be other antibodies produced, and there will be another type which is known as a memory B cells, which are produced so that if any secondary infection is happening with the same pathogen, it can remember the pathogen and can act accordingly. So bone marrow is not a site of B cell maturation in all species. For example, in birds, first it was found this bursa fabricus, which is it is a pouch-like structure out of the intestine, and then in other species like in other animals like cattle or sheep, this bone marrow, the B cell maturation takes place in spleen, fetal spleen or pairs patch. In rabbit, it occurs in appendix. This is the bursa fabricus. You can see here is a pouch-like structure. Here in birds, the B cells get matured. After that, there is spleen. Spleen, as I told, it is another secondary lymphoid organs, and it will also have that uh, structure, and it will contain these resting B cells and dendritic cells. It will filter blood. It will trap blood-borne antigen, and if there is you are cutting this spleen, then there will be systemic infection with various bacteria. So this one was week one's class. It's has taught. So any week, if you want, I can discuss with you any specific week or any specific topic. If anyone needs you want. Hmm.
Maybe about the TLRs, something like that, that receptors related.
TLRs. Okay, I am going to TLRs.
The weak cells.
So pattern recognition receptor, as by the name you can understand, there are certain receptors which can recognize some patterns. So whose pattern they can recognize? From the pathogens which are entering into our body. So this is Charles A. Jenner. So he was the person uh, who have been studying this, and this was first found in Drosophila. Some protein was found, toll protein, which was shown to activate the adaptive immunity in Drosophila. And this after that they found that similar to Drosophila, some uh, like some homologs are found there also in humans. So this was another paper in which they showed, this is the paper in which they showed there is a human homolog to the Drosophila toll protein which activates the adaptive immunity. And this paper showed that there are, if there is any kind of defect in LPS signaling, then in mice they saw that there are some mutation in some gene, TLR4 gene, which is causing some defect in the TLR signal, in the LPS signaling. So this is the innate immune response in Drosophila. You can see there is some fungi is entering into the into this into the system. Then there is some activation of the antifungal peptide genes, which are occurring through some receptors, which is recognizing some antigens. And this was named as toll. Toll obviously means some gate through which it is entering. So this is another famous personality from Japan, Professor Shizuo Akida, and most of the genetic studies regarding this PRRs are done by his group. So innate immune response in Drosophila, uh, there are cells for defense and there are antimicrobial peptides. The cells for defense includes the hemocytes and fat body cells. They are the phagocytic cells, just like our macrophage or DCs or natural killer cells. There are antimicrobial peptides like drosomycin, dipterosin, atacin, which are produced. Then these are the microbial sensors in the Drosophila that are found. They are like peptidoglycan recognition proteins, gram-negative binding proteins. They are may basically involved in recognizing the peptidoglycan from the bacteria, or they can also recognize fungi. They can recognize certain molecules from gram-negative bacteria. Then from gram-positive also bacteria, they can recognize some peptidoglycan molecules. So this is the downstream signaling of the sensors in Drosophila. There are certain serine proteases, so known as GRAS, and they trigger some proteolytic cascade which cleaves the protein spatzel. Then there is the spatzel uh, creates a homodimer, means two spatzels join together. Then there is dimerization of toll and it stimulates the antimicrobial response. So toll is central to defense in Drosophila. However, toll itself is not a pattern recognition receptor. So it mainly controls the dorsal ventral patterning in Drosophila. So see, it's a toll pathway in Drosophila, but we will now come about the pattern recognition receptor which are present in our body. So there are different pattern recognition receptor like toll-like receptors, Regai-like receptor, nod-like receptor. Like means it is having some what to say, some similarity with the toll, so they have named this as toll-like receptors. So toll-like receptors can recognize bacteria, fungi, viruses on surface and in endosomes of cells to induce pro-inflammatory cytokines and type 1 interferon. And there are Regai-like receptors which also can sense RNA of RNA viruses and induce pro-inflammatory cytokines and type 1 interferon. Then there are nod-like receptors, which are also intracellular sensors, which can recognize PAMs, which are nothing but pathogen-associated molecular patterns. So these are pattern recognition receptor. They can recognize some patterns which are present in the pathogen known as pathogen-associated molecular patterns. They also induce pro-inflammatory cytokines and involved in processing IL-1 family cytokines.
So how does it actually, like, what part of the like bacterial micro, like what as does it actually attract? I mean, like sensors,
Uh,
right? Like what
Sorry, I
part of
cannot
it?
actually hear you.
Oh, 1 minute. No, I'm just asking. Like, can you hear now?
Yeah, yeah.
So I'm just asking, just what exactly, which part of the microbe does it exactly bind to the receptor? Like,
Like this toll-like receptor, there are different kinds of toll-like receptor, TLRs. So suppose some TLR can recognize suppose some virus is entering, it can be RNA virus, it can be a DNA virus, it can be any kind of virus. So some TLR can recognize that RNA of that particular virus
okay.
or
But
some,
is it like it is present inside the body, right? So how does it exactly find it out?
present inside the body means.
Like it is the virus is actually present inside like the RNA is present inside the virus, right? So like it
But
is
when a
like
virus is
uh,
entering into our cell, it will release its genome
okay.
on our cytoplasm. So then only it can recognize that. Or if some bacteria, for example, TLR4 recognizes bacteria LPS. So LPS it can recognize, but LPS will be present on the outer surface of the bacteria. So it can recognize that molecules, LPS molecules, which are present outside. So, for example, some TLRs are localized on the cytosol and some are on the endosome. So, whatever are inside our cell in the endosomes, it can recognize, as I told, a virus. When it is entering only, it can like release its genome. So that endosomal ones will recognize that, but whatever it can recognize from the outside, like the LPS, it can recognize from the surface. So some these receptors also will work downstream only because it also recognizes that RNA of the viruses. So depending on the location of these receptors also, it depends what they will recognize. For example, here you can see these are the different locations. Like in the plasma membrane, this TLR4, as I told, TLR4 is localized. So TLR4 uh, can recognize the LPS, which it can directly recognize whenever there is a entry of any kind of bacteria into the cell. But this endosomes one, which can recognize only after there is a release of that what to say RNA. Then there is this TLR, these three plasmamembrane TLRs can recognize, for example, this TLR 6 and 2 can recognize some lipopeptide. So it will directly, whenever there is an entry of the pathogen, it can recognize that. So that time, like during the direct entry only, it can recognize these three TLRs. But this endosomal TLR only can recognize after the pathogen is entering inside. So whether that molecule is exposed on the surface or it is means inside, it depends which TLR is recognizing what. So here you can see like these are the DCI, MDA, LGP2. These are also not present on the surface. These are present in the cytosol. So only they can recognize after a pathogen is entering into our body. It is releasing its genome. Then only this can recognize that. It cannot recognize when it is binding to the receptor. But these three, Dectin 1 can recognize directly the pathogens when it is trying to enter in. So here also you can see this. This is the signaling pathway through which all these pattern recognition receptors uh, can activate certain pathways and can kill or destroy the pathogen. So there are different kinds of toll-like receptors. So you can see 1 to 10, um, and it is a single pass transmembrane protein is reaching leucine rich repeat. So they are here you can see, as I told, TLR 1 2 can recognize triacyl lipopeptide, TLR 2 6 can recognize diacyl lipopeptide, LPS can be recognized by TLR 4, TLR 5 recognizes flagellin, TLR 11 recognizes uropathogenic bacteria. So all these are present on the cell surface. So directly it can, and most of these you can see are bacterial, which can recognize the bacteria. But whatever are recognizing the virus are present inside. So these are the 5 types of the TLRs which are present on the surface. So it can directly interact with all the exposed things which a pathogen possesses. But here you can see the endosomal one. It can recognize, this TLR3 can recognize double stranded RNA. This TLR7 or 8 can recognize single stranded RNA. TLR9 can recognize CpG DNA. So all these uh, can recognize different kinds of molecules. It can recognize fungi, it can recognize bacteria, it can recognize the virus. Depending on the surface molecule, each TLR will recognize what kind of pathogen is that. So here again you can see these are the different kinds of things, different kinds of molecules which are recognized by the TLRs. So there are two types of signaling. One is mediated dependent signaling and another is strip dependent signaling. So mediated dependent signaling, here you can see mediated dependent signaling. Signal is coming to mediated, which is converging at this place, and mostly all of this activate this NF-kappa B signaling pathway, which induces the production of this pro-inflammatory cytokine. And there are also certain transcription factors, IRF3 and IRF7, which will go and bind to the go to the nucleus, bind to this DNA, and will induce the production of type 1 interferons. And mostly all these pathways converge at a same place. And there is a chief dependent signaling pathway. Well, here it also it activates the TBK1, then which activates the IRF3 or 7 pathway, which is activating the type 1 interferon. So mostly these are seen in macrophages dendritic cells, and in mediated also macrophage dendritic cell and plasmacytoid dendritic cells.
Hello.
Yes.
Yeah, no, my connection was lost. That's why. Please continue.
Okay. So you can understood what I told.
Yeah, yeah, yeah. Now it's pretty good. That one.
Okay.
Yeah.
So here is the mediated dependent signaling pathway. Yeah. So here mediated, you can see this many TLRs can activate the or mediated. It can, the signaling pathway goes to the mediated pathway, and here it can either activate IRF7. It can bind to IRAC proteins. It can which lead to the activation of NF-kappa B pathway, then production of inflammatory cytokines. Then this IRF7 will go, as I told, and production of type 1 interferon will be there. Then chief dependent signaling. Trip dependent signaling also. This TLR4 mainly is activating. Here also we saw TLR4 is activating this trip dependent signaling, and sometimes TLR3 also. So this TLR3 and TLR4 are mainly activating this strip. These are the adapters through which the signaling occurs. Then there can be two pathways: MAP kinase pathway, which is producing these inflammatory cytokines. This TLR4 can also induce this MyD88. Then there will be NF-kappa B signaling. Then there is IRF3, which also it can activate. So you can see all these are converging at the same place almost, mostly either to the NF-kappa B or through the IRF3 or IL-7, or it is through the MAP kinase pathway that these are all activating the immune system or the pro-inflammatory cytokines of the interferons. So these are the different kinds of TLR, and they are having different adapters, as 3 and 4 will have TRIF, and mostly all the other will have 8, MyD88. So these are the type 1 and type 3 interferons, which are inflammatory mediators are there, as well as type 1 type 3 interferon. Type 1 interferon consists of IFN alpha and IFN beta. Then there will be type 1 interferon again will induce certain interferon inducible genes, which we known as interferon stimulated genes, which we know call as ISGs. Mostly these are IP10 adantes, and many are there. Then there are type 3 interferons, which consists of IFN lambda, which consists of 3 members. Apart from this, there are other proteins like IL-1 beta, IL-6, TNF alpha, histamine, serotonin, other vasoactive amines, icosanoids, there are peptides, bradykinin. So, role of type 1 interferons in antiviral responses. Here is the how the type 1 interferon is inducing antiviral response. Here you can see many pathways lead to the production of this type 1 interferon. There can be certain molecules which we know as OAS, or oligoadenylate synthase. So from here, it is producing this OAS, and it is leading to the production of type 1 interferon. There can be production of this RNase L, which can degrade the viral RNA. There can be PA, PI, P1 kinases, which will activate the EIF2, which will inhibit the translation of proteins. There are MX proteins that are which increase resistance to virus like influenza. There are NK cells which directly can kill the viral infected cells. Then there is obviously adaptive immunity. There will be CD4 and CD4 plus 8 T cell responses. There will be increase in antibody. There will be increase in MHC1 expression. So that. So here you can see if there is any kind of polymorphism or there is any kind of mutation in any of these signaling molecules, it can lead to different kinds of diseases. For example, here you can see TLR4, some D299 is changed to G, means the aspartic acid is changed to glutamine. Then there is a like disease, and here you can see there will be like a decreased response to inhaled LPS, decreased atherosclerosis, increased gram-negative bacterial infection, increased severity of atopy, decreased airway hyperreactivity. So these are the different symptoms which can happen when there is a polymorphism or mutation in the TLR4. Then here you can see IRAC4, if there is a stop codon at the 287 or 293 place, there is an increased gram-positive infection. Then there is increased response to LPS if the CD14 is changed. So you can see different signaling molecules which are, if there is any kind of mutation, it can lead to diseases. Then there is one NEMO, which different kinds of polymorphism has been found in this NEMO factor, which is important for the NF kappa B signaling. And here, people who are having this are increased to viral and bacterial infection. And there is I kappa B alpha also, which is having any this mutation. Serine is changed to inosine. Uh, then there is impaired T cell memory. So here also some examples are given, which are
Actually,
yeah.
structure. So basically, that CD14 will be present together with the TLR,
It
or
is a
is it like?
co-receptor kind of co-receptor. Sometimes with the receptors, you need some co-receptors so that there will
Yeah.
be binding affinity with the ligand, or some structural modification is needed, which is provided by this co-receptor. So whole structure is important for it to function. If CD14 is also mutated, the function of the TLR4 will also be hampered.
Okay, okay. So only CD14 is like that, or many kinds of CD40, some other kinds of numbers, like other kinds are always as co-receptors or can be separate also.
No, no, co-receptor can be separate. Many are there, CD. Like CD4, we tell about CD4
Yeah.
T cell, CD8 T cell. Apart from
Mm.
that, it is having many types of co-receptor along with it. So all the co-receptor also should be in place to activate the T cell or the B cell, the T cell receptor.
Mm. Okay. Okay. And this IARC is also together. Only I've heard in my subjects all only one signaling occurs, then only IRAC will also come. Is it like that?
One signaling means?
Like if you, for example, it binds, mean the next process step takes place, or is it like
Huh.
permanently it will be like this only, this
No, not
process?
the permanently. First ligand will bind to the receptor, then it will activate the mediated. Then mediated will recruit this IRAC from the cytosol. It will come and bind to this mediated. There will be then phosphorylation events. Then only it will, the downstream signaling process will take place. So in any step, if it is not happening correctly, then the whole signaling process won't work.
Okay.
So these are the polymorphisms and linkage to different kinds of diseases. I told different mutation in the amino acids or the nucleotide. Similarly, here also, if there is any in the adapters or in the IRACs. So these are for cancers. They are the different kinds of molecules which can, if suppose in a cancer, one TLR2 or TLR4 is not working properly, we can give a agonist. Agonist means something which is mimicking the function of that particular uh, molecule. Like it will help in the function. Like if a particular TLR2 or TLR4 is not working, then this agonist will help in the uh, like it will do the function of the TLR2 or TLR4. This OM174, it is in the phase 1 clinical trial. Then these are the different kinds of molecules.
So is it
Uh,
like chemically synthesized or it's natural?
yeah, chemically synthesized.
Okay.
It will be similar to the
Mm.
structure of the TLRs, or so that it can mimic the its function.
Yeah. Okay.
So another one is the this imiquimod, so the TLR7, and is launched for papillomavirus infection. These are the different agonists and antagonists. Agonist we need whenever there is a something is not functioning, we have to mimic that function. And antagonist means we have to, if there is an excessive function or excessive activation of some molecule, then we have to give some antagonist. So there should be like so that activity is suppressed and there is no disease which is occurring due to the hyperactivation. So these are the different kinds of antagonists for TLR4. Similarly, here is a list for bacteria viral infection that target the TLRs. Because as I already told, not hyperactivation and not hypoactivation, both are bad. You have to have a homeostasis, and there should be some positive regulators as well as some negative regulators to control the balance. So this is all about the TLRs. So the uh, pattern recognition receptors. RLRs will come in.
So toll-like receptors and toll pathway, that is, the both are same, or like what is their difference? Or they are just having that one?
Toll is the protein which was found in Drosophila, and it was found that it is activating some immune signaling pathway in Drosophila.
Yeah.
Then
Mm.
some similarly structured protein was found in our human body. Like its structure was similar to
Mm.
that toll protein, so they named it as toll-like receptor. So somewhat
Okay.
structure was similar to the Drosophila protein. So then comes the pattern recognition receptors or the RLR. So this was one paper which showed that this Rigai is RLR means Rigai like receptors. So this Rigai is an antiviral molecule and it can uh, induce some innate antiviral responses. These are the RLR sensors, Rigai, MDA5 and LGP2, and these are having different kinds of domains. A card domain is there, then helicase domain is there, transmembrane domain is there, and CTD domain is there. So these different domains have different functions. Like obviously, card domain, which is mainly this caspase activation and recruitment domain. Its full form is. So this is having a like a function of oligomerization and recruitment of other molecules. Then this helicase domain is there, and there is there is like RNA recognition domain. So all these are having different kinds of function. All these domains are having. So these RLRs can recognize RNA viruses. These are cytosolic compared to some TLRs which were on the plasma membrane. These RLRs are in the cytoplasm or cytosol because they as so they can also recognize the RNA which are released by the virus when they enter the cell. So here you can see this is Rigai and this is MDA5. They Rigai can recognize Newcastle disease virus, vesicular stomatitis virus, Hendai virus, and MDA5 can recognize encephalomyocarditis virus. This Rigai can recognize triphosphate RNA, means the virus which are having this triphosphate RNA is recognized by this RIGI, and MDA5 can recognize this double-stranded RNA. So here you can see these are the CARD domain. It is helping in oligomerization, and there is um, activation of this molecule IPS1 or MAPS, which is again activating the same pathway as I told as in case of TLRs, IRF3 or 7, which is going into the nucleus and activating type 1 interferon, or it can be NF kappa B pathway, which can activate the pro-inflammatory cytokines. So here is the basically the structure. Here is the RIGI, this is the helicase domain, this is a C-terminal domain, and here you can see it is interacting with the 5' triphosphate RNA of the viruses. So these are the 7 classification of Baltimore, which divides it into different classes like virus classes like double-stranded DNA, single-stranded DNA, double-stranded RNA, positive single-stranded RNA, positive negative single-stranded RNA, reverse transcriptase like transcribed single-stranded RNA, then reverse transcribed double-stranded DNA. So all these you can see VGI and MDA5 these are recognized by these two molecules, RLRs. So during infection, uh, it also happens that uh, our own RNA can activate our RIGI because it cannot distinguish. Obviously, it can only see that a pathogens whether a pathogen is coming or whether a host RNA is coming. It like if the similar sequence is coming like a triphosphate RNA, it can recognize them. So sometimes what happened in our case, sometimes host cellular RNA can be misprocessed or mislocalized. There can be disturbances of cellular RNA homeostasis. There will be different kinds of non-coding RNA which are produced in our body, which can be recognized by this RIGI. So there can be mutation as well in RLRs, and there if there is some any mutation and if there is activation, then constant activation will be there in absence of virus infection. Then these are the different syndromes like Singleton-Merten syndrome, Aicardi-Gouttier syndrome. So here you can see this Aicardi-Gouttier syndrome. There will be long hairpin RNA which are derived from our inverted. Some repeats will be there in our body, which are known as allo elements. They will be inverted repeats, like similar repeats will be repeated many times, and sometimes long hairpin RNA will be generated from that, and they will activate the mutant form of this MDA5. So this will activate the signaling pathway, and this will lead to this disease. And also some mislocalization of some RNA or wrongly processed RNA can also activate these RLRs. Then again, these other molecules are there, like adenosine deaminase acting on RNA1 or ADAR1, if deficient in humans and mice, cause chronic type 1 interferon production in inflammatory pathology. In breast cancer, the tumor microenvironment results in increased production of the non-coding 5' triphosphate containing this RNA. So you can see not even in virus cells, sometimes in cancer cells also there can be triphosphate containing this RNA which are produced, which can activate this RIGI. So RIGI is not only specific for viruses, any kind of triphosphate RNA it can recognize, be it your host RNA, be it your cancer cells RNA, or it can be of any kind of pathogens RNA. So this is a signaling pathway. It is activating IPS1, then TBK1, then similarly IRF3 or NF kappa B or IRF7. These are the regulation of this RLR pathway by different kinds of proteins and non-coding RNAs which are regulating this pathway. There can be some post-transcriptional modification of RIGI and MDF5 like ubiquitination or anything. Then these are the mutations which are occurring in these RLRs and it can lead to different diseases. Here, as I told, like RLR agonist that comprise poly-IC. Poly-IC is a compound which can also activate these RLRs. Then the 5' triphosphate double-stranded RNA, stem loop RNA can induce cancer cell apoptosis and immune responses against tumors. So we can treat this uh, condition with like RLR agonist can be made, which will activate that pathway and can it has been shown that it is good for cancer cell apoptosis and immune response against tumors. So if you put poly-IC, this higher weight poly-IC, it was found that anti-cancer effect of ionizing radiation is enhanced and following upregulated FAS expression is responsible to FAS cell-induced apoptosis. So it can be an option where you can give the RLR agonist. There can be activation of these pathways. It can be which is helpful for treating cancers. Then this SLR14 is a RIGI agonist which has been observed to induce anti-tumor response in immunogenic or non-immunogenic tumors, and it can have a powerful immune adjuvant of immune checkpoint therapy. Then there are other molecules like nanoparticle delivery of some natural ligand of RIGI, like if we put a from outside, if we are putting some phosphorylated double-stranded RNA into a pancreatic cell line, then it will increase the RIGI signaling, and there can be uh, less tumor progression. Then again, here you can see M8, which is a modified sequence of RIGI agonist. It is a RIGI agonist and it consists of a hairpin 5' TPP RNA, and it is showing that cell death related and damage associated molecular patterns on tumor cells, and it can also be acting as a therapy for different kinds of cancers. So you can put, if there is a low level of RIGI which is causing some disease, then you can put some agonist, can activate the signaling. If there is high expression of this RIGI, then you can put some antagonist which can control the situation and can help in the like reduce the progression of the disease. So these are the, as already shown, these are the different kinds of RLR mediated therapeutics which can actually be used. Then we come to the NLRs. So NLR is another type of pattern recognition receptor, and like toll like receptor which was first found in Drosophila, NLR were first identified in plants. So plant cell, there will be certain genes which are known as R genes, which can actually recognize this microbial and parasitic pathogen, and these genes are known as NLRs, and therefore after that uh, it was named as NLR in our case. And apart from plants, homologs of these NLRs are also found in vertebrates as well as the primitive organisms like sea urchin. And so from it is the function of NLRs are evolutionary conserved like the toll proteins. Like it helps in the host defense, be it any kind of vertebrate or it can be any kind of organism. So in human, in our NLR is composed of 22 proteins, and there are at least 33 NLR genes in mice. So these are the different types of NLRs which are present in our body. So NLR means not like receptors, nucleotide binding, oligomerization domain. So here also different kinds of domains are there which are having different functions. Like these are the structure and the function of the NLR. There are different NLR, node 1, node 2, NLR P1, NLR P3, NIP5. So this node 1 and node 2 can induce the production of these cytokines through the NF-kappa B and MAP kinase, and this node 2 induce the production of the inflammatory cytokines. Same similar function. Then the ligands, as I told, it can be different kinds of ligands. Like mostly here we can see are the bacteria which are recognized by this NLRs. Then this NLRP1, it is also recognizing the bacteria. Then it induces apoptosis or caspase-1 dependent cell death. Then this NLRP3 is an inflammasome. We call it as an inflammasome because it consists of many kinds of, it is a complex like structure and it induces caspase-1 dependent IL-1 beta and IL-18 production. It forms NLRP3 inflammasome along with ASC and caspase-1. The ligands, these are the different ligands, and mostly it can be anything. Like here apart from the bacteria, we can see other like UV radiation, beta amyloid protein, anything can activate this inflammasome and there will be high inflammation upon activation of this inflammasome. Then there is this NAIP3 which controls replication of bacteria independent of caspase-1. There is ligand includes Legionella. Then this is the receptor, flagella of Salmonella typhimurium, uh, which can recognize this. And mostly the function of this NLR is activation of apoptosis or this IL-1 beta or IL-18 production, and some like NOD2 and NOD1 can activate NF-kappa B MAPK kinases. So here you can see NAM and NAC are the ligands. Obviously we know are present in the bacteria. So these are recognized by this NOD 1, NOD 2. So here you can see these are the different kinds of uh, ligands recognized by these two molecules. This is the signaling process. It is activating this NF-kappa B pathway or to the MAPK pathway, the inflammatory cytokines. It is activation of the inflammasome, as I told, NLRP3, NLRP1, different kinds of NLRC4, different kinds of inflammasomes are there, and it can activate this caspases which will cleave the pro IL-1 to IL-1 active form. The types of inflammasomes include this NLRP1, and these don't only recognize the viral or the bacterial DNA or RNA, but also some DAMs. DAMs are nothing but damage associated molecular patterns. Means whenever there is a damage in our body, some amount of things will be released from our cells. So this includes this ATP, sodium, potassium, efflux. Then there is production of ROS. Then if there is a disruption in the mitochondrial membrane, then they will release some molecules which are also recognized by this NLRP3 inflammasome, which will activate the signaling. So as usual, if there is as as usual, if there is any kind of mutation in the NLR, there will be different kinds of diseases. For example, in NLRP3, you can see this Muckle-Wells syndrome, familial cold, autoinflammatory syndrome, neonatal onset. Different kinds of diseases are occurring whenever there is a mutation in this NLR receptors. So NOD2 is known to be a cause of this Crohn's disease. Here you can see NOD2 is there. There is more production of these pro-inflammatory cytokines. So reduced NOD2 dependent expression of bactericidal alpha defensins in PANH cells. Here then there is impaired recognition of the bacteria by this NOD2 and clearance of the bacteria. So there will be enhanced production of IL-1 beta, which will cause a more and more of inflammation, which can cause this Crohn's disease. So again, these are the different kinds of diseases which are occurring upon mutation in the NLR receptors. These were all about the bacteria or mostly the RNA sensors. Then comes the DNA sensor. So DNA sensors, especially TLR, TLR9 is known as the DNA sensor. So it can recognize some CpG DNA which are found in this microbial or microbes. So here you can see these are the papers which showed like DNAs can be also recognized by these toll-like receptors. First reported DNA sensor was this dye or DNA dependent activator of IRFs. It is a IFN inducible gene and was capable of upregulating the expression of IFN1 by NF kappa B. Similarly, here also you can see these are merging at this NF kappa B or the IRF pathways. So these are the different kinds of DNA sensors which are found. Dye is one of them, first one. Then there are different kinds of adapters, like MyD88 is adapter for the TLRs. Their here adapter is mainly Sting. Also, these are the names of the Sting, Meta, Aries, etc. Then Sting also interacts with TVK1 and activate IRF3. Apart from this, there are other DNA sensors apart from dye like AIM2, IFI16, DHX9, 3641. These are the different kinds of DNA sensors which are being found over the years. So these DNA sensors can recognize any kind of foreign DNA and they can detect the perturbation in homeostasis also and can activate the signaling process. They induce type 1 and pro-inflammatory cytokines. They can also induce programmed cell death. It depends on the methylation status of DNA, the localization of DNA molecules, sequence of cytotoxic DNA, association of histones and non-histone chromatin binding proteins with cytotoxic DNA molecules. The DNA sensors are these are the cytosolic DNA sensor, IY16C gas, DDX41 dye, PQBP1M2. These DNAs one cleave this single-stranded DNA and double-stranded DNA in extracellular space. While TLR9, these are the nuclear DNA sensors which are residing in the nucleus, and it senses TLR9 and DNS2 senses the extracellular DNA translocated in the endolysosome. So TLR9, as I told, senses some CpG molecules found in the pathogens. Here you can see the signaling process. This is the endolysosome. There is this TLR9, there is dimerization, then through the MyD88 adapter. Then there is dimerization of this IRF1, IRF4. Then it is activating this IRF7, which is activating the proenzyme type 1 interferon. Then again in another site, it can activate the NF-kappa B pathway, which is leading to the production of this pro-inflammatory cytokines. So it can sense different microbial pathogens like HSVs, HPVs, VZV, CMV, many different kinds of pathogens you can see. Another one is C-gas. C-gas is a cytosolic DNA sensor, and it is activated by DNA binding, which triggers conformation changes that induce enzymatic activity. Here DNA is binding to C-gas, which is going and binding to STING. The C-gas is changing to C-GAMP, and it is binding to STING, which is a localized adapter. Then STING interacts with this TBK1, then again that IRF3 pathway and NF-kappa B pathway it is activating. So cytosolic DNA is efficiently degraded by some TREX1 molecule, which is the exonuclease. Exonuclease means it can cut from the ends only, and endonuclease means it can cut from the between. So there are certain cytoplasmic double-stranded DNA which are sensed by our immune system as non-self nucleic acid. So it can activate uh, this signaling process upon sensing this cytoplasmic double-stranded DNA. This AM2 is another inflammasome which comes under this. So M2 has different kinds of domains, and it is as inflammasome. I told most of the inflammasome function is to activate this caspase pathway, then activating the IL-18 or the IL-1 beta cytokines. Then another is IFI16, which senses cytoplasmic as well as nuclear DNA. It also activates this IRF3 and NF-kappa B pathway. Some DNA sensors are also there which can also sense RNA. For example, IFI16 and DIE can also sense viral RNA apart from viral DNA. It can sense both virus as well, like RNA virus as well as DNA virus. And ZDNA or RNA is recognized by the Z binding domain which is present in ZBP1 or DIE. So it can lead to pyroptosis. There are DNA ligand structures. For example, here you can see a structure which is greater than 40 to 80 base pair, and these can be recognized by the C-gas, M2, etc. And they can be recognized by your immune system as non-self, and it can activate the signaling pathway. So these are the DNases which can get activated and create autoinflammation. As I already told, sometimes our body will recognize our own self DNA or RNA as pathogens and can activate these pathways, and it can lead to different kinds of diseases. For example, here it is leading to type 1 interferonopathy, meaning that the excessive production of type 1 interferon is occurring and there is more and more amount of inflammation. So this STING and CGAS pathway, similarly to the other pathways, it can some agonist or antagonist can be produced which can activate this. So I think that is all uh, for the pattern recognition receptors. If any more doubt is there, then I have 15 minutes. I will clear it. Any other doubt is there?
No,
Okay.
thank you.
Ah, thank you. So if any kind of other doubt is there, you can tell me because I have still 15 minutes to discuss any kind of slides or any kind of MCQs. Hi Kalpana, if you are having any doubt, you can ask or any kind of topic you want me to discuss. Okay, then thank you. If you know what, you're not having any doubt, then thank you. This is the last class. If any kind of question is there, you can post in the portal.